Resolution and detection of nucleic acids. Electrophoresis. Electrophoresis is the movement of molecules by an electric current. So you can see a negatively charged side and the arrow going toward the positively charged side. Nucleic acid moves from the negative to the positive pole. Nucleic acids have an overall negative charge, so when we load nucleic acids on the negative charge, it's going to be repelled from like charges and migrate toward opposite charges because opposite charges attract. So if we have negatively charged DNA and we load it near the negatively charged electrode, it's going to naturally want to migrate through the gel toward the positive charge. When DNA is applied to a macromolecular cage or gel, such as agarose or polyacrylamide, its migration under the pull of the current is impeded. So when we put DNA into a gel, the gel itself is molecular, and the DNA particles will have to work their way through this gel mesh. Imagine you walking into a party and it's wall-to-wall people. So if the room was empty, you'd be able to just walk right through to the other end of the room. But because it's wall-to-wall people, you now have to bump and work your way through this huge crowd to get to the other side. It's kind of like what happens with DNA or any molecules that are trying to get through a gel. There's a lot of resistance to getting through that gel. So the movement of molecules is impeded in the gel such that molecules will collect or form a band according to their speed of migration. So those that travel through really fast are going to be visible at the bottom, and those that don't travel as fast are going to be up at the top. The concentration of gel buffer will affect the resolution or separation by size of fragments. Slab gel electrophoresis can have either a horizontal or vertical format. Sample is introduced into wells at the top of the gel. So if it's the horizontal one, you would put the sample in here, again by the negative charge, because DNA has a negative charge, and we always want to run it to the opposite end. One of the things that we say commonly in molecular is run to red. Run to red means that you are going to orient the gel so that the DNA is supposed to be migrating through to get to the positive charge, which is usually a red electrode. Here we can see a vertical. In this one, we would load it at the top and they would run down. So more about how DNA migrates through the gel. So DNA strands that are super small can get through a lot faster. So imagine if you come to this party with some kids. Now kids are shorter, they're smaller, they can migrate through that crowded room a lot faster, as opposed to an adult, which may take a little bit longer to get through because they're bumping into and trying to get through all of those spaces. So when we have different size bands or different size fragments of DNA, it usually means that they are different sizes. Now if we're talking about resolving DNA specifically, when we talk about this, DNA is going to have a consistent negative charge throughout, and when we load the DNA at the top, it's going to migrate through at a predictable speed. And it's predictable based on the size of the fragment of DNA that we're trying to resolve. So DNA fragments that are shorter, and you can see down here it says 50 something base pairs, those that are shorter will be able to go through faster than those that are larger won't be able to migrate through as fast. The percentage of gel also makes a difference. So if you have 20 people in a crowded room versus 100 people in a crowded room, if we have a higher concentration of people, it's going to be harder to get through. Same thing happens when we're looking at the percentage of gel. Most gel, when resolving DNA, is going to be from 0.5% to 5%. So here you can see what it would look like separating bands at 2% versus 4% versus 50%. And if we look at that last base pair, that 50 base pair, it'll go a lot faster through if it's only a 2% gel versus if it's a 5% gel. It'll go through a lot slower. The other thing we can mess with is the voltage applied. So higher voltage is going to get it through a lot faster. But sometimes we don't want DNA to go through really fast. If bands are really close together, we can adjust the percentage of the gel, and we can also adjust the voltage applied to the gel. And if we run it for a certain amount of time, we can mess with the time. So we can run it for longer, which will resolve fragments a little bit more, as opposed to running it for a shorter amount of time. So all of these factors we can adjust to try to separate out bands of DNA, especially if the two bands or two anticipated sizes are going to be pretty close to each other. We mess with the percentage of the DNA, the percentage of the gel, the voltage applied to the gel, and the time that we run it through the gel. And that can allow us to separate out these bands and visualize them according to what we're looking for. Very large DNA molecules are separated by pulsed field gel electrophoresis, or PFGE. And there's 4 main systems of these that exist. But just kind of going back here, you can see these big blobs at the top. These are pretty large molecules of DNA. So if we are trying to look at different fragments within this big blob, then we would have to use something like pulsed field gel electrophoresis. So the types of PFGE: field inversion gel electrophoresis, alternating positive and negative poles. So here is a positive pole, positive down here, negative. So these will then alternate positive and negative poles. And the whole goal is to switch up the current to tweeze out the DNA bands. So DNA is going to be pulled in one direction and then pulled in another direction and slowly work its way up toward the outer edge, and that allows us to separate out those larger strands. Transverse alternative field electrophoresis, a transverse angle reorientation of poles on a vertical angle. So here you can see that process happening. So there's different poles which will allow this to kind of move apart in a zigzag pattern. Contour clamped homogeneous electric field, alternating polarity in electrode array. And you can see that at the bottom. And rotating gel electrophoresis, rotating gel with fixed poles. And you can see that there. So the gel actually rotates. Again, the whole purpose is to apply alternating currents to try to tweeze away these molecules that are pretty close together because they're so large. Polyacrylamide gel electrophoresis, or PAGE, is usually used for separation of proteins. So if we're going to do a protein separation, we typically do this, but it can be used for DNA occasionally. So with PAGE gels, typically we have SDS as the main buffer, so that becomes an SDS PAGE gel electrophoresis. With proteins, now if you remember from biochemistry class, proteins are made up of amino acids. And amino acids have very specific side chains, and those side chains give the protein its overall structure. Every protein has a particular isoelectric point and has these side chains that will give it more of a positive charge, more of a negative charge, a hydrophobic, a hydrophilic. It gives some different features to the protein. So proteins and SDS page, we can really mess with a lot of things. Not only are they separated by size, but they're also separated by charge. So where if you run a gel in a certain pH environment, it will run one way, and if you run a gel in a different pH environment, it'll run a different way, because proteins and their side chains, they're all kind of work in a different way. So that's kind of the next level. We're just kind of focusing on DNA and how we would separate strands of nucleic acids out on a gel. So acrylamide is used in combination with a crosslinker, like methylene bisacrylamide. Synthetic consistent polymer, so it's consistent throughout the gel. Then there's polymerization catalysts. We can use APS or TEMED, or light activation. Resolves one base pair difference in a 1 kilobase molecule, so a 0.1% difference. We also have capillary electrophoresis, or CE. This separates solutes by charge-mass ratio. Capillary gel electrophoresis is used to separate nucleic acids. So this is what it looks like. You have buffers on each side. There's a negative, there's a positive end, and then you're going to have the sample being sucked up by capillary action, and as they go through, they're separated by the charge and mass ratio. And then they're picked up by a detector. Thin glass, fused silica, capillary, 30 to 100 centimeters multiplied by 25 to 100 micrometers internal diameter. Linear or cross-linked polyacrylamide or other linear polymers are used for sieving. Separation is based on size. CGE is more rapid and automated than slab gels. A lot more commonly done in the clinical setting. Run at a high charge per unit area, and then the electrokinetic injection of the sample. Electrokinetic injection, nucleic acid fragments to be separated are drawn into the mouth of the capillary with a transient positive charge. Once collected, fragments migrate through the capillary to the positive pole. Buffers are really important, especially when we talk about those side groups with proteins, but focusing more on nucleic acids, we want to make sure that we have a consistent pH throughout. And when we add nucleic acids, it can mess with the pH. So we want to make sure that the pH remains the same. Buffers prevent a change in pH. So they carry current and protect the samples during electrophoresis. Trisborate EDTA, trisacetate EDTA, trisphosphate EDTA are used most often for DNA. 10 millimolar sodium phosphate, buffer used for RNA. Buffer additives can be used to modify sample molecules. Formamide, urea, denaturing agents. Horizontal or submarine gels are usually, but not always, agarose gels. So here is usually the black electrode, which has the negative charge. Here's the red one, which has the positive charge. We load our samples into the wells here and then turn it on, and they're going to migrate in this direction. Now, the reason why people who work with DNA are constantly thinking run to red is because if you happen to orient this in the wrong way, where you put the wells next to the positively charged one, what's going to happen is they're going to run off the gel, and you just lost all the DNA that you just extracted. You're going to have to do the extraction process all over again. And in some cases, you did a lot of work to get that DNA to how you want it, and now you want to evaluate it, and you can't because it just ran into the buffer solution, and now it's floating in the buffer, and it's impossible to isolate from there. So very important to always remember run to red. Vertical gels are usually, but not always, polyacrylamide gels. So you would run it in the top, and then it would be drawn down toward the positive electrode. Combs are placed in the gel before polymerization to leave wells in the cast gel for sample loading. So here are different types of combs that can be used. Basically, that's going to help make these indentations in the gel. When you pour the gel, it's going to be liquid because we heat it up and we mix it together, and then we pour it, and then we put these combs in there so that when it cools off and solidifies, those wells are left behind. Use the proper gel concentration for the sample size range, so 0.5 to 5% agarose, 3.5 to 20% polyacrylamide. Use the proper comb and gel size to accommodate the sample number. Run a molecular weight marker of known fragment sizes. So we want to have a marker where we know this particular band means 100 base pairs, this one means 200, this one means 300, and so forth. So that way when we do run our DNA out and we have different bands that are separating out on the gel, we can compare it to the marker of known band sizes to determine what our band size is. We mix the sample with tracking dye plus a density agent, and then we pipette the gel into the wells. We connect the gel bath to the power supply, confirm a current or amperage is passing through the gel. Typically, we can determine that when we see little bubbles starting to form inside the bottom of the gel container. Detect bands by staining during or after electrophoresis. Ethidium bromide is used commonly for double-stranded DNA. This is a carcinogen. It's a known carcinogen, and we have to be very careful with it. Cyber green or cyber gold for single or double-stranded DNA or for RNA. We also have cyber safe, and that's a safe molecule that doesn't have any carcinogenic effects to it. Silver stain, more sensitive for single or double-stranded DNA, or for RNA and proteins. Silver is toxic, so we have to be careful with the silver. And you can review the summary.